Okay, so ethanol can cause cellular injury in a couple of different ways. It can be due to nutritional deficiencies or it could be due to oxidative stress. So nutritional deficiencies occur when alcohol is ingested long term. Deficiencies in magnesium, vitamin B6, thiamine, phosphorus, and folic acid can occur. And all of these adversely affect the brain and the peripheral nerves. And this might be something that you see with Wernicke's encephalopathy, peripheral neuropathies, or Korsakoff psychosis. In addition, the folic acid deficiency is the key thing that is causing fetal alcohol syndrome in pregnant women who drink alcohol. So what occurs here is that ethanol alters folic acid homeostasis by decreasing intestinal absorption of folate, increasing liver retention of folate, and increasing the loss of folate through urinary and fecal excretion. Okay, but that was the nutritional stuff. And we also said that oxidative stress due to the metabolism of alcohol can cause illness. So we'll need to think about how alcohol is metabolized in the body. And that's done through 3 enzyme systems. The main workhorse is alcohol dehydrogenase, and then we have the microsomal ethanol oxidizing system, MEOS. MEOS is dependent upon cytochrome P450 enzyme for work and able to work. And that piece of the metabolism is what causes the oxidative stress. The other enzyme that works to metabolize alcohol is catalase, and catalase actually takes the breakdown of hydrogen peroxide and breaks it down further into water, releasing a hydrogen ion. So, like we said, the main workhorse is alcohol dehydrogenase, and alcohol dehydrogenase is basically releasing hydrogen ions, right? A hydrogen is released when alcohol dehydrogenase turns alcohol into acetaldehyde and bound to a compound called NAD+, or nicotinamide adenine dinucleotide, and it forms nicotinamide adenine dinucleotide plus hydrogen. Okay, and so that was at the cytosolic level. The next step is at the mitochondrial level is when ALDH2 breaks down acetaldehyde into acetate. So we're going to have another release of a hydrogen ion and to form the acetate. Acetate is then released as ketone bodies, fatty acids, and amino acids into systemic circulation. The breakdown of acetic acid is used actually in the Krebs cycle for aerobic metabolism. Okay, so that's the whole breakdown of the metabolism of alcohol. And like we said, the problem lies in the microsomal ethanol oxidizing system. So what happens there is that you are breaking down NADPH, losing a hydrogen ion. That hydrogen ion is then ionized, having a loose electron charge, which causes oxidative stress to occur. Remember when we, if you went back to cellular injury, just talking about basic hypoxia, there was some discussion about this. So you probably remember this. The reactive oxygen species or oxidative stress that forms can damage the lipids, proteins, and DNA. The peroxidation of lipids occurs when there's destruction of unsaturated fatty acids. So fatty acids have these double bonds between carbon atoms and are vulnerable to attack by these oxygen-derived free radicals. And this causes excessive production of hydrogen peroxide and hydroxyl radicals in the mitochondria that damage lipids, proteins, and mitochondrial DNA. The lipid peroxidation, actually, right? So that it all went back into that circle of free radicals, and unstable electrons or unpaired electrons that are unstable, ending up causing either apoptosis or necrosis and cellular death, depending on the situation.